this lesson, we're going to look at the cell cycle. Now, one caution here, oftentimes people will use the terms cell cycle and mitosis interchangeably. That's actually not technically accurate. The cell cycle describes the entire life cycle of the cell. This whole thing down here is the cell cycle. Notice that mitosis is a piece of it, okay? So just understand that. There are 3 steps to the cell cycle. Step 1 is interphase. Now, interphase has its own steps as well. So this part of the cell cycle here that goes from here all the way around is interphase. Okay. Step 2 is mitosis, and again, mitosis has its own steps. These you're familiar with probably from middle school: prophase, metaphase, anaphase, and telophase. Step 3 is cytokinesis. And again, students often get confused because they think of cytokinesis as part of mitosis. Cytokinesis is not a foregone conclusion. You can actually have mitosis but no cytokinesis, and some of your body cells will do that because they need to have a cell that is long and fibrous and needs all parts of it to be under control, so it needs multiple nuclei to do that so that each nucleus controls a part of the cell. So your muscle cells will do that. So cytokinesis is not necessarily always have to be present in all kinds of cells, but it usually does. And it usually actually takes place at the same time as telophase, but it doesn't have to. Now, can you have mitosis without interphase? If you do, you probably have a cell that's cancerous. So there needs to be this period of interphase between cell divisions as much as possible. All right, so step 1 is interphase. All right, now interphase, remember, of the cell cycle is this whole part here, and you can see its 3 parts. So first part of interphase is the G1. Okay, and the G1, and by the way, all during interphase, the cell is carrying out normal cell function. In the G1, the cell's growing. It's duplicating all of its cellular components, all of its organelles. It does not duplicate, though, the chromosomes at that point, or the DNA at that point. The second part of interphase is called the S phase, and the S stands for synthesis because now it's synthesizing brand new DNA. It's duplicating DNA. The last part is called the G2. And the G2, it continues to grow, but it's getting ready now for cell division. There is a checkpoint right here in the G1 called the G1 checkpoint, and that's a point where the cell monitors certain levels of hormones within the cell. There's one called cyclin, for example, which might be good for your honors project, that kind of, if it's present in a certain amount, then the cell will continue from here into the S and G2. If it's not present, then the cell will hold for a little bit and continue to grow until enough cyclin is present. And that's kind of what regulates interphase, is all these chemical signals. I've included here a picture of what the cell looks like after synthesis. So this is really what a cell looks like at interphase during either late S phase or during the G2, because you've got duplicated cellular DNA here. But it looks like what you kind of picture a cell to look like. The only thing you can't see is all the other little organelles here. All right, the second step of the cell cycle is mitosis. For the moment, let's just ignore this picture and let's just go right here. Now, here's interphase, right? Okay, and remember that interphase is not part of mitosis. We've dealt with interphase. Mitosis actually starts here at prophase. Okay, so part 1 of mitosis is prophase. And because this is an animal cell we're looking at, you have these 2 new organelles that pop up called centrioles, and the centrioles produce these spindle fibers, and these are long protein structures that are going to help us to pull apart chromosomes. Some other features of prophase: notice that the nuclear envelope begins to dissolve, the chromatin condenses down and coils up into chromosomes, and notice that the centrioles move to opposite sides of the cell and the spindle fibers kind of stretch out between them. Step 2 is metaphase. Remember that meta means middle, Okay, so in metaphase, notice that all the chromosomes are lined up in the middle or along the equator of the cell. Each chromosome is connected to the spindle fibers. And notice that our centrioles are on opposite sides of the cell at this point. So we're getting ready to kind of play tug-of-war between this centromere and this centromere over here. Step 3 is anaphase. And during anaphase, the centrioles start to shorten the spindle fibers and pull on those chromosomes, so they separate at their sister chromatids, or excuse me, at the centromere. The sister chromatids get pulled apart and start being pulled to opposite sides at the centromere. The final step is telophase. And during telophase, the DNA starts to unravel and become chromatin again, and the nuclear envelope starts to reassemble. Spindle fibers disappear, centrioles begin to fade, and we've finished mitosis at this point. Now, a quick little word of caution. First of all, what you're going to be expected to do mainly is not so much be able to put the names of these in order, but if you're given a series of pictures, we would expect that you can put the pictures in order and kind of describe what you see happening in each of those steps based on the picture. Also, I wanted to contrast, let's turn our attention down here, plant cell mitosis and animal cell mitosis. Remember we said back in the last unit that these centrioles that form are not present in plant cells. Those are animal cells only. So you'll notice in a plant cell at prophase, or metaphase or anaphase, whenever, you're not going to see any kind of centriole pulling on the spindle fibers. The spindle fibers do the pulling themselves. The final step is cytokinesis. Now, remember I said before, cytokinesis often happens at the same time as telophase, but it's not a foregone conclusion. It can happen, it usually does, but some cells will go through mitosis but not do cytokinesis. Cytokinesis is where the cell actually splits. Okay, so here's the last step. Cytokinesis, here you see the cell splitting into two cells. So we've finished off telophase, or we're in the middle of telophase here, and see how the cell starts to bend in toward the middle and the cells kind of pull apart here. That's what's happening at cytokinesis. Eventually what's going to happen is this side is going to meet this side as they pull together, and you're going to pinch off into two new cells. Again, that's what animal cell cytokinesis looks like. Plant cell cytokinesis is kind of different. Notice here at the end of anaphase is where we are here. As telophase proceeds in this cell, notice that you start to see these little dots forming along the equator of the cell. Remember that plant cells have to form a new cell wall boundary between them. So what these little dots will become, they'll start to fuse together and assemble into new cellulose molecules. This is called the cell plate. Let me write that out here. This is the cell plate. Cell plate. And the cell plate, that's an E there we go. The cell plate will form between the two daughter cells that we've formed, and you will end up with two cells that are now divided by a brand new cell wall. Now, at this point, the cell has completed its whole life cycle. These cells will enter interphase and go back into the G1 and continue to grow, and then once they reach their maximum size, they'll start their S phase and their G2 and so on, and this is how the cycle proceeds. Notice also, when you look at these cells that are produced are identical to the original cell. This is an asexual reproduction process. It's a natural cloning process that you're seeing happening. So the cells are just copying themselves, and the daughter cells that result are identical to the original cell.